The pigmentation of the cuticle can vary in flies in general and fruit flies specifically, with some flies having a darker pigmentation and some flies having a lighter pigmentation. Obviously, among other reasons, this could affect a fly's ability to blend in with its background. In Drosophila melanogaster, the wild type or the typical pigment is a tan colored body, but there are mutations in a number of genes which can alter this wild type phenotype and produce a mutant phenotype pigmentation hue. The gene ebony is a gene for an enzyme involved in the conversion of the amino acid beta alanine into a number of products such as dopamine or histamine. And this conversion is important to generate histamine, which functions in the eye, or dopamine, which affects some aspects of behavior, or the production of cuticle pigmentation. And so thus the mutants ebony possess a darker pigmentation than the wild type, in addition to a number of other effects. This is a pleiotropic gene which has multiple effects on phenotype. And so thus ebony flies not only have a darker pigment in the adult cuticle, their pupil cases are actually lighter in color, and they can have a number of other altered phenotypes, such as altered behavior and a longer development time. This mutant phenotype is incompletely dominant in that heterozygotes have a pigmentation which is intermediate between the pigmentation of the wild type and homozygous mutant conditions. So for example, in a cross in which all of the males were ebony and all of the females were wild type, the next generation would have all heterozygous individuals whose pigmentation was intermediate between the wild type and ebony colorations. In the F2 generation, if the F1 was allowed to cross, one would see not only a 1 to 2 to 1 ratio in the homozygous for the wild type allele to heterozygous to homozygous for the ebony allele, but also a 1 to 2 to 1 ratio in the phenotypes. In cases of incomplete dominance in the F2 generation, the phenotype ratio and the genotype ratio are the same, where 1/4 would be wild type, half would be the intermediate heterozygotes, and 1/4 would be ebony. Humans have a homologue of this gene whose genetic notation is AASDH. It is a gene in the acyl coenzyme A synthetase family.